So, DNA is a vital molecule for every cell. It's the blueprint of life. But on occasion, mistakes or changes occur in DNA and in the code, causing various problems for cells. And any difference or change in DNA is referred to as a mutation, right? And so these mutations or changes can be caused by a number of things. Sometimes DNA polymerase makes errors or mistakes during replication. Sometimes when our DNA molecules become damaged and they're trying to be repaired, they can make the mistake even worse, right? And then we've got things known as mutagens. And mutagens are um, elemental factors or conditions that can result or induce changes in our DNA. We've got physical, right? And a good example would be radiation. So ultraviolet or UV radiation, we all know can cause mutations in our DNA. In fact, that's why we wear sunscreen to protect our cells, to protect our DNA from UV radiation. That's also why we tan, and that's why some people who have darker skin pigmentation have a decreased chance of experiencing these UV radiation-induced mutations. We also use UV radiation to sanitize surfaces and to clean drinking water because the UV radiation does cause changes in DNA, which can cause problems for the microbes that are in water, right? You want to remove them before you drink it. Chemical compounds can cause mutations. It can be mutagenic. So nicotine, in cigarettes could be mutagenic. Other compounds like DDT, right, which is an insecticide that caused a lot of problems, that's mutagenic as well, can cause changes in DNA. And then we've got biological agents like viruses, they can actually induce changes in our DNA resulting in mutations. And these mutations or changes in our DNA would alter the proteins that they code for, right? And so some of these mutations can prove to be very disastrous. Some mutations are actually considered neutral and they don't really result in significant changes. And then on occasion, very rarely, these mutations can be considered beneficial. Now, there are different types of mutations, and I've got some of them listed right here, right? So maybe this is the original code, right? And maybe a mutation occurs and we've changed the C to a T. We changed cytosine to thymine. This would be a substitution mutation. Now, if this is the original sequence, and let's say that we, right here, between the G and the C, we were to insert an extra nucleotide, like we've done down here, we've got an insertion mutation. And what you see is the original code was TACGCC. Now that we've inserted that A, everything is moved down right. And so these insertion mutations can have a significant influence on the way in which the code is actually read. In the same way that we take letters and we combine them together to make words, DNA made of these letters, the T's, the A's, the G's, and the C's, is not read in a continuous, like one letter at a time, but they're actually grouped together in sections of 3. And we'll talk more about that when we get to translation. And then we've got deletion mutations, right? So maybe we just remove a nucleotide entirely. And again, this can cause major problems because everything would then shift to the left, right? And these little groupings of 3. So these are the 3 main types of mutations. And insertions and deletions, these mutations can be very serious because they influence the rest of the code downstream. So, right, since we've inserted this A, everything down here has been kind of shifted over. And if we were to delete the A right here, everything is going to shift to the left. So everything downstream from that mutation is affected. And we call these frame shift mutations. Now, where these mutations occur is also very important to consider. Mutations that occur in our somatic cells will only influence us, right? So if I were to spend too much time out in the sun and I end up with some form of skin cancer that resulted from mutations in my skin cell, that's not something that I can pass on to my offspring, right? Because the only cell or cells that have anything to do with my offspring would be my gametes, right? Now, however, if a mutation were to occur in my germ cells, right, the cells of the testes, the cells of the ovary, that mutation could then be passed on to the offspring. So it's kind of a weird thing to think about. If a mutation occurs in a somatic cell, a body cell, it's only going to influence or affect that individual. However, if the mutation occurs during meiosis, or the mutation is present in the cells that undergo meiosis, then the mutation would be passed to the offspring and might not necessarily influence the individual producing those gametes.